The broadcast is now starting. All attendees are in listen-only mode.
Hello everyone, and welcome to the latest installment of the HDSA Research Webinar Series. Today I'm happy to announce we have Dr. Jenny Morton from the University of Cambridge joining us, and she'll be presenting today on sleep and circadian dysfunction in Huntington's disease. Before Jenny gets started, just want to go through a couple housekeeping issues with everyone online. We will have time dedicated at the end of Jenny's presentation to answer your questions. You can send them at any time during the presentation, so just to send the question, go to the control panel that you should see on the GoToWebinar site on the right-hand side of your screen, and in the panel it says question. Just type your question, it'll be, and click send, and it'll come right to us, and we'll ask Jenny the question on your behalf at the end of the webinar. Just of note, 1 week after the webinar is broadcast, we will be recording this and posting it on our HDSA national website. So to access this presentation, just go to hdsa.org/researchwebinar. In addition to this webinar, you can view any of the old webinars that have occurred. And finally, I just want to remind everyone of our upcoming schedule. On May 22nd will be Joe Giuliano, who's the Director of Clinical Operations from the CHDI Foundation in Princeton, New Jersey, will be joining us here in the New York office to give everyone an update on the Enroll HD observational trial. In June, instead of having a research webinar, we will be instead having webcasting our research forum from our national convention in Jacksonville. And the topic there, we'll be talking about speakers such as Michael Hayden and Jeff Carroll and Ed Wild will all be talking about latest developments in HD drug development programs. And finally, if you have any research webinar topics or suggestions you'd like us to cover, please email us at researchupdates@hdsa.org, and we'll do our best to try to find a good speaker to cover that topic for the community. So without further ado, I'd like to introduce Dr. Jenny Morton. Dr. Morton is a professor of neurobiology at the University of Cambridge in the United Kingdom. She has been working in the HD field for a long time, since around 1991, where she's been a leader in HD animal model development, particularly mice, and now most recently in the development and characterization of our large animal model, the HD sheep. So many HD patients have difficulties in getting a good night's sleep and with control of their daily circadian rhythms. And it's hypothesized that improving sleep in HD patients would have beneficial effect on cognitive and emotional disturbances. So what we've asked Dr. Morton to do is take us through the work that she and others are doing in HD animal models to better understand sleep and circadian dysfunction in Huntington's disease. So with that, I'll turn it over to Jenny, and we'll just hand over presentation rights to you, Jenny, and you have the floor. Thank you, Jenny.
Excellent. Thank you, George. So it's a great pleasure to talk to you about sleep and circadian timing and Huntington's disease, because it's something that's interested me for about 10 years, long before I really realized how much of a problem it was for patients. So today I'm going to go through, I'm not going to present a huge amount of data, but I'm actually going to go through what sleep and circadian abnormalities are and how they might affect an HD patient. And a lot of the evidence we have has come from mice, because they're much easier to manage than humans, as you will see. So circadian first. What does this mean? So circadian means approximately a day. And this comes from the fact that the Earth rotates once every 24 hours. And pretty much on average half the time we spend in the light and half the time we spend in the dark. So you have a light period and a dark period, and the period of the rotation of the Earth is pretty close to 24 hours. And this governs a lot of what we do. So most of us will go, yes, well, of course, we sleep. So sleep down here, I've got Snoopy here, sleeping, we sleep at night, and we tend to be active during the day. But there are many other rhythms that are governed by the circadian period, and we have 24-hour periods of neurotransmitter release, body temperature, our activity, which is not quite the same as sleep, and I've distinguished them here, and I'll go into this in more detail in a minute, electrical activity in our brain, hormone levels, our cognitive performances. There's a cycle for each of these rhythms, and it's governed by our circadian timing. The origin of the circadian timing is actually in the brain, and there's a tiny nucleus at the base of our hypothalamus. So this is a picture of a cross-section of a rat brain, and this is the front of the brain, this is the back of the brain, and this is the hypothalamus. It's an area that controls many of our automatic functions. And right down here, you can see, if you take a section through here and flip it onto the side, you can see the side, the coronal section of a brain. Right down here is this tiny little nucleus called the suprachiasmatic nucleus of the hypothalamus. We call it the SCN for short, for obvious reasons. And this little nucleus generates its own 24-hour rhythms in response to light that comes in through the retina. It's a very clever mechanism that determines every 24 hours this nucleus generates electrical activity that dictates our circadian rhythms. So it's called the master clock or the master controller. And you can see here that I've got an example of something called a periodogram that I'm going to go into in a minute from a rat, and it's blocked into day and night, and you can see that this animal is active in this period, not active in this one, active here, and he has a 24-hour rhythm. We know that the SCN governs circadian rhythms because if you lesion this area, you stop being rhythmic. So if you have a lesion of both nuclei, these tiny little nuclei in the brain, in a rat, you eliminate all rhythms, and you can see that the animal becomes dysrhythmic if you've got an SCN lesion. So what difference does this make? Well, it makes a big difference because we were working on R62 mouse model of Huntington's disease, which some of you will be familiar with. If you're not, I'm just going to run quickly through what the R62 mouse model is, because these mice lose their circadian rhythms. So they're transgenic mice, and they carry a fragment of the gene with the mutation that causes Huntington's disease. So in the gene, there's a little fragment of the gene that's variable, and so in a normal person, it varies between typically 8 and 24, can be as much as 36, and people will be neurologically normal. Longer than that, you have Huntington's disease. And this fragment, a long fragment, is enough to cause disease in these mice. So the mice are born normally, and then they start showing progressive symptoms. First, body weight changes, then they start showing movement abnormalities, and they have cognitive deficits, until by the time they're 15 weeks of age, you can see that they look abnormal. They've got hunchbacks, their fur's not very well kept, they're not very good at looking after themselves, and they have brain pathology. Now, the real advantage of using mice is that you can control them, and in particular, you can actually measure circadian rhythms, because we're very influenced by other things in terms of our circadian biology. So it's all very well to say that we have a 24-hour drive from the sun, but we also have families, we have work, we have jobs, and these things modulate rhythms in humans. In mice, now mice are actually nocturnal animals, so whereas humans have a low activity in the dark and a high activity in the light, mice are the opposite. So they have low activity during the daytime, they sleep during the day, and they're active at night. But otherwise, the mechanisms that generate their rhythms are very similar to humans. And we can take a mouse and we can put it in a box by himself. It's very important that they're by themselves. And there are infrared beams in this box, and every time the mouse moves across the infrared beam, you get a mark of activity. So if the mouse is very active, you'll get lots of activity. If the mouse is asleep and quiet, you'll get very little activity. So we can record the locomotor activity for as long as we like. We can put the mice in boxes like this for 60 days. So here's a recording where the mouse has been in the box for 60 days, and each one of these recordings across is 48 hours. It's called a double plotted actogram because we collect the data continuously for 60 days, but we plot it twice. So the first time we plot it across the whole width, and then we repeat it so you can see that every 24 hours there's a change in activity in this particular mouse. So this mouse, this is the dark, beg your pardon, this is the light, this is the dark. This mouse wakes up here when the lights go off, is very active at night. When the lights go on again, goes to sleep. This is the same as this period. Sleep, wake, sleep, wake, sleep, wake. And you can see that this mouse has a very strong 24-hour rhythm that's driven by the lights on. And what we did, and we did these experiments quite a long time ago now, but I'm going to show you the original data because it's still very, very reasonable, is that we have normal mice called wild-type mice who have a 24-hour rhythm. And we have R62 mice who also have a nice clear 24-hour rhythm when they're young. 6 to 8 weeks of age is a young adult mouse. Now these two actograms differ. This one actually looks less organized than this one. This is typical. There's big individual variation even in mice, so that doesn't matter. What's important is that when the mice get older, and in the lower panels you can see a normal mouse who's now 4 months old, 14 to 16 weeks, and you can see he's still got a very nice clear 24-hour rhythm. But the R62 mouse, the rhythm has disintegrated, and he doesn't have a nice rhythmic periodicity to his day. Even though the lights come on here, he should be asleep. The lights go off there, he should be active. This mouse is active and then inactive pretty much regularly all through the day. So he has periods of activity, periods of inactivity, but they're not blocked nicely around a circadian clock. So this was the first finding that we had, that there was something wrong with the circadian regulation in Huntington's disease from this mouse model. With my collaborators, Roger Barker and Carrie Hurlbunk from the Brain Repair Centre in Cambridge, Roger and Carrie actually put ActiWatches onto humans. And I'm showing you these examples of human ActiWatch data to show you really that it's not as clean as the data that you get from mice, but it's still interesting and tells us something important. So in the top panel, this is a control subject who's a normal person, doesn't have Huntington's disease. This person is active during the day, so as with the mice you get periods of activity during the day. Sometimes he's more active, sometimes he's less. Then at the reported bedtime, the activity subsides massively. There is still some activity because people wriggle around and they move a lot at night, and the watches pick up any activity. Then you can see when this person gets up and becomes active again. The second graph shows the same sort of thing, activity for a patient. And this is a patient who's very early stage and very relatively unaffected. And this person still has a nice circadian rhythm. They wake up, they're active during the day, they go to bed at night, low activity. So there's a periodicity. Every 24 hours this person goes to bed. The bottom panel is a patient who is much more severely affected, still active and still independent, but you can see that this person's activity profile is not as normal as the one above. If you look at this data in the same way I just showed you the mouse data, a double-plotted actogram, you can see this better. On the left-hand side, these are all normal subjects, and these data again came from Roger and Anna Robbins' study. normal subjects, you've got nice periodicity. Every 24 hours these people get up, every 24 hours they go to bed, and each of these is a different subject. So you can see there are differences, but the similarity and the rhythms are the same. On the right-hand side, you can see a selection of early-stage Huntington's patients, and there's much more variability in the actograms of these patients. We did an analysis of this, and this graph shows what's called a night-day ratio. Now, you don't have to know exactly what that is, but effectively it's the amount of activity in the nighttime expressed as a percentage of that in the day. Now, you can see that what I've called a non-carer, and I'll come back to that in a minute. So this is a neurologically normal person. Relatively little, about 10% of their activity, total activity, happens in the night period. Patients, about 25% of their activity happens in the night period. Now, this is not chorea, so this we can exclude chorea from the watches. This is actually just generalized activity, not relating to abnormal movements. In fact, the patient group that Roger used didn't have chorea. Now, I've called these non-carers because we had two groups of neurologically normal people. We had people who had nothing to do with Huntington's patients who are normal. We had HD patients who are gene positive, early symptomatic, without chorea. And we had carers. There was a significantly worse effect on the patients, and this was mimicked in the carers. So carers had almost as bad circadian rhythms as the patients did. So this is a significant knock-on effect of the circadian rhythm deficit that the patients had is seen in the carers. And we thought this was important because it's not just the patients who are affected, but also their families, and anybody who looks after HD patients will know how devastating this can be. So how important are sleep-wake abnormalities per se? Well, for normal people, they're very important. We have evidence from jet lag, we have evidence from shift work, and we have evidence from other neurological diseases that disrupting your sleep is bad for you. Jet lag and shift work in particular, there's a lot of study done on understanding the deleterious effects of having your circadian timing artificially imposed or dysregulated. So people who cross the Atlantic Ocean, people who work nights have side effects of their sleep disruption that are known to be unfortunate. People with neurological disorders don't have the abnormal timing that shift workers do, but they still have serious effects. And in fact, in Alzheimer's disease, this has been studied very well, that there's a breakdown in circadian rhythm. This is, again, some double-plotted actograms from a healthy control, an Alzheimer's patient with mild dementia, and an Alzheimer's patient with moderate dementia. And you can see, again, that there's a breakdown in circadian rhythmicity. And in fact, in Alzheimer's disease and Parkinson's disease, treatment of sleep and rest activity is a targeted, focused subject for both of the research communities in these diseases. So they recognize that circadian disruption in Alzheimer's and Parkinson's disease is important. In fact, in Alzheimer's disease, it's been suggested that the major reason for institutionalization is, or going into a home, is their breakdown in circadian function. So just to summarize that a little bit, for a normal subject, sleep deprivation has side effects. The side effects that are classical for a normal person with sleep deprivation: anxiety, depression, poor decision-making, poor memory, concentration lapses, irritability, and a lack of interest in sex. These are things that are classically associated with sleep-deprived normal people. Alzheimer's disease, Parkinson's disease, these are some diseases where it's known to, circadian disruption is known to be a symptom. I've added Huntington's disease into that because I think we should consider that it might be as important as it is in these other diseases. And certainly Huntington's disease patients frequently have irritability, and towards the, as their disease advances, often have cognitive deficits, in particular executive function, making poor decisions, for example. So if they also have sleep-wake deficits, then there's no reason why they shouldn't have all of these side effects from their sleep deficits, as well as any irritability, for example, that comes from carrying the Huntington's disease gene. So I think it's something that we should consider, that sleep deficit, whether or not it's a side effect of the disease or whether it's part of the disease, may actually make symptoms worse. And this raises the possibility that actually treating the circadian disorder might improve neurological function. This is something that I've suggested. There's no evidence for that yet because there have been no tests done to test that hypothesis. Okay, so I'll come back to this in a few minutes. What I want to do now, though, is segue into sleep, because sleep is not the same as circadian rhythms, and it's very confusing because circadian deficits are sleep-wake deficits controlled by the SCN. Now, sleep is a circadian behavior, but it's actually not generated by the SCN. Sleep is a complex behavior with multiple components, and it's controlled by other parts of the brain. So it's a behavior that's under circadian control, but it's the SCN is just the master controller that controls sleep as well as the other circadian deficits. I'm not going to go into what controls sleep, but I am going to mention that there are three main components of sleep that are probably regulated by different or generated by different parts of the brain. And these are slow wave sleep, rapid eye movement sleep, and wakefulness. So waking up is actually a mechanism that is generated from a different part of the brain than going to sleep. Now we measure sleep, and we call the sleep architecture the components of this slow wave sleep, the rapid eye movement sleep, and wakefulness. Altogether, we call it the architecture, and I'll show you why in a minute. And you measure this by electroencephalography, EEG. Some of you will have had EEGs. Few of you will have had EEGs for sleep because this is called polysomnography, but EEG measurement gives you a pattern of what your brain's doing while you're hooked up to a machine. And if you go, let me go back for a second, so you can see this subject here. I took these photographs off the web, so I attribute to them to the person who made them. But if you look at this man here, he's asleep, and you can see he's got EEG electrodes just on the surface of his skull, but he's also got other electrodes. So he has electrodes on his chest, he has electrodes to his muscle, and this is the polysomnography, so it's multiple electrode sleep recording. And what you get from polysomnography is a complex set of data, and I've shown you again some data from Roger Barker and Anna Goodman and John Schneerson, whose group did the recordings. And this is raw data from a normal subject. You can see that there's all sorts of things measured. So down here it's got oxygen concentrations, apnea, whether or not people stop breathing, whether they snore, whether they're aroused. These are the sleep stages, which I'll come back to, whether or not their limb movements, whether or not they have periodic limb movements, and their body position. So you get a lot of information from these recordings. What I'm interested in is these details between here and here. And these show you the stages of sleep. And they're labeled by W for wake, 1, 2, 3, and 4 for the 4 stages of sleep going down to deep sleep, number 4, and REM, the rapid eye movement sleep, that are characteristic of these stages. So when a normal person goes to bed, they are awake at the beginning, then they go sequentially through different stages of sleep until deep sleep. And this lasts typically for about 60 to 90 minutes, and then about 90 minutes after you've had a sleep, you go into a period of REM sleep. And this is when, if you're woken up from the sleep, people typically report dreaming. And you cycle through these cycles of sleep all through the night. This person's had a little bit of arousal there, they're woken up for a little bit, and then gone back to sleep. And at the beginning of the night, you have more deep sleep. You can see the periods get shorter as the night goes on. And towards the end of the night, you have more REM sleep. But these cycles, these periodic cycles, happen all through the night. And you can see why it's called architecture, because it looks a bit like a city. So this is sleep architecture, and this is a normal subject. Now, when Roger and Anna did the Huntington's disease patients, and this data has been published, so you can look at original graphs from all of the other patients as well, because they're in the papers that Anna and Roger published. The architecture of the HD patient is disrupted. And what's interesting is this is an early stage patient, and they have very disrupted sleep architecture. This patient also had multiple arousals, so they woke up quite a bit, they snored a little bit, and they didn't have periodic limb movements. But what was most interesting was the sleep architecture was abnormal. And if I put the normal, if I just paste the normal subject's sleep plot on top, you can compare the difference between this normal one and this HD one. So this patient was not severely affected. He was still working, still fully functional, and didn't actually report bad sleep. So the many of these subjects didn't feel that they were sleeping badly, but their brains had poor architecture, and so whatever is generating sleep stages and wakefulness is abnormal in this person. I'll summarize this very briefly, because the critical things are that there is less REM sleep and less slow wave sleep, that they had frequent awakings and disrupted sleep cycles and multiple transitions between these states. But they didn't show much abnormal sleep behavior, which is what you might expect if you had very disrupted sleep. They didn't report particular sleepiness. So their brains had abnormalities, but the patients didn't report having sleep abnormalities. Now, I put a caveat on here because the numbers of patients that we looked at were low, and this is one of the real problems with studying Huntington's patients, is that half of the patients were actually taking drugs, and many drugs that people take affect sleep. So drugs that people take for, I mean, even caffeine affects sleep architecture, and so this is not an ideal study. The other thing is that several of these patients were actually depressed, and it's known that depression causes abnormal sleep. So only 3 of the patients that we looked at were drug and depression free. Importantly, all 3 of those patients showed abnormal sleep architecture, so I think it's a real phenomenon. But we need more research done on this. We need more studies done to understand how these sleep changes, what these sleep changes are, and how important they might be for Huntington's. There was another paper published about the same time from Alexandra Dur's lab, and she also showed that there were rapid eye movement sleep disturbances in Huntington's disease. But there have been surprisingly few actual studies in Huntington's patients in sleep clinics. There's also relatively few studies in animals. In fact, I have just had a paper, we've just had a paper accepted for publication in Brain showing that there are significant changes in sleep EEG in R62 mice, and it will be interesting to see whether or not the changes that we're seeing in the mice will be found also in patients. We do a different analysis on sleep in mice, but it should be possible to do a similar analysis on human subjects if a study is ever carried out. So we've got two different things that are going on here. We've got circadian abnormalities and sleep architecture abnormalities, and these are interconnected because they are both circadian behaviors. It's very hard to separate them in humans. So the question is, I've just gone back to lumping them all together as sleep-wake deficits. Can we do anything about them? Without any more studies, there aren't drugs that are HD specific that will treat them. Is there anything that can be done if you have a sleep-wake deficit? Well, I think yes, there probably is. So what I've done here is I've summarized what the SCN does in terms of its clock, that it modulates all of these things: endocrine system and peripheral nervous system, rest activity cycles, sleep-wake cycles, feeding when you eat is circadian driven, and even body temperature is circadian driven. So all of these are circadian functions. And I think we can affect rest activity cycles. There is already evidence in HD patients that not being sedentary improves performance, that patients who don't take any exercise and are very passive tend to decline a little more rapidly than patients who are active. Patients who are in work, who have a routine to their lives, also, the longer people stay in work, the better off they seem to be. And people who do, so I put rest here as something, as a focus, that regulating your rest activity cycles with exercise, sensible regimes, may actually improve your rest activity cycles, which have a knock-on effect on your circadian rhythms. Sleep-wake cycles being separate, good sleep hygiene is important, and I wrote an article recently for HD Buzz that was based on a review that I'd written. There's no reason why Huntington's patients should be an exception to a normal person's rule, that we tend not to sleep very well. We play with electronics too much, we have our phones on, we work at night, we watch television. We're not, the 21st century human is not very good at looking after their sleep hygiene. So improving your sleep hygiene, i.e. sleeping in a dark room, not having your phone on, not texting in the middle of the night, not watching television, and not going to bed too late and getting up at a sensible time, will probably improve all of our sleep-wake cycles, including Huntington's patients. And I haven't talked about it, but there is another circadian oscillator that's called a food entrainable oscillator, and this is regulated separately from sleep and separately from rest activity cycles. And so feeding time may actually be very important for regulating your circadian output as well. So having regular and healthy food seems obvious, but the regular may be more important for Huntington's patients than it is for a neurologically normal patient. And the other thing I put on this slide that has never been looked at in Huntington's disease but has been looked at in Alzheimer's disease and Parkinson's disease is the possibility of using bright light therapy to improve your circadian rhythms. So altogether there are a number of things that you might be able to do that will improve your general circadian physiology, even in the absence of a specific Huntington's disease effect. So exercise, regular life, good sleep hygiene, good eating, and sleeping in the dark and having light in the morning is probably not going to do you any harm and may actually will improve your circadian biology. So to summarize, I think that we do know that sleep disturbance is problematic for Huntington's disease patients, and that sleep disruption in people who are neurologically normal has negative effects on daily living. So whether this is a chicken and egg thing, it's not clear. My guess is that some of the sleep abnormalities in Huntington's disease patients are because of their disease, that it's actually part of the disease. And so an HD patient would be sensible to try and minimize the effect that normal people have, the negative effects of their lifestyle on their sleep, because it may exacerbate any Huntington's disease related effects. But it's not known if sleep-wake deficits are a symptom of Huntington's disease or just a consequence of having them. We need more research to understand that. We also don't know if drugs used to treat sleep disturbance in normal people work as efficiently in people with Huntington's disease. That's not been studied either. But I think it is possible that sleep disruption in Huntington's disease patients might well exacerbate symptoms, and so improving sleep hygiene, improving circadian lifestyle are likely to be beneficial for Huntington's patients, as they would be for patients or for normal people who have disrupted sleep. But it may be more important for Huntington's disease patients because you're starting from a disadvantaged background, as it were. Okay, so that's all I wanted to say. I mean, I have plenty of other things to say, but I wanted to leave time for questions. So I'd just like to thank the people that have really helped me establish the work we've been doing in Huntington's disease. Mick Hastings, Liz Maywood, and Joanne Chesham, who've worked on the circadian rhythms in the mice. Roger Barker and Carrie and Anna Goodman, who have done the human studies, and Steve Dunnett and Leslie Jones from Cardiff, who actually, we did a molecular study that got me thinking about circadian rhythms in the first place in the early 1990s, and the people from my lab without whom any of this work could be done. So Sandal's working on the EEG in mice. Nick Parentos has started working on EEG in the sheep, and we're hoping that using a large animal model, will give us more insight into the mechanisms underlying the changes in sleep and circadian rhythms. Okay, so George, that's all I wanted to say, so I'm really happy to answer questions if there are any.
Thank you, Jenny. That was a great presentation. Yes, if anyone has any questions, please feel free to ask them. Now, I'll just start off and give people a time to ask a few questions. I'm interested, Jenny, about the SCN and your data from the HD mouse, the R62, and I was just wondering if anyone's ever looked at. It seems to me that there's maybe a non-specific neurodegenerative effect, since you're showing these circadian and sleep deficits in many neurologic disorders like AD and PD and Huntington's. I'm wondering if anyone's ever looked to target mutant Huntington specifically in the SCN, like with a knockdown approach, to see if lowering Huntington in just that little brain region of the hypothalamus has a beneficial effect.
Yes, I don't think anybody has. They have targeted Huntington to hypothalamus, but not treatment for it. It's an interesting question because it may be that region, some of the knockdown improved. It's a bit like that. Huntington changes are all over the brain, so I don't think that would treat Huntington by itself. It may delay some of the symptoms, which would be important, but I don't think that's not the cause of Huntington's disease, that little region.
Okay, thanks. So I have a few questions here, Jenny. One is, do you have any suggestions for caregivers whose sleep is being disturbed by a patient other than sleeping in a different room?
You know, I think if you talk to anybody whose husband snores or whose you know, children are disruptive and you want to work, I don't think there are easy alternatives. My husband once my children slept very badly when they were little and screamed all night, and my husband once slept on the ground in the garden because he had a big talk to give the next day, and that was the only way he could get sleep. And it's very disruptive when somebody you live with doesn't sleep very well. And yeah, I wish there was an alternative, but sometimes having a sort of bed holiday is probably the only answer, that you go and actually decide to sleep somewhere else for a week and catch up on your sleep if you're a carer.
Okay.
It's not a satisfactory thing, but it's sometimes to get your head back to normal, it might be worth trying.
So there's another question. It's with a keen interest towards your sheep data. How can you elaborate more on that, and if you're seeing any kind of sleep abnormalities in your Huntington sheep?
No, we haven't got to the stage of looking at sleep abnormalities in the Huntington sheep, because we've now got sheep that we can do in Cambridge that we can measure EEG recordings, but we haven't yet been able to do this in the Huntington's disease sheep. I'm hoping that in the next year or so we will be able to do this, because obviously trying to figure out the actual mechanism of what's happening here in an animal means that we can try therapies, and if they don't work, that's not ideal, but we don't lose much, and we learn a lot. So yeah, I'm hoping that in the next year we will have, we've got the EEGs working. We've just not done any in the Huntington's disease sheep yet.
Okay.
So that's coming soon.
Thank you. There's another question about inquiring if any research has shown the benefits of melatonin or other sleep medications, such as maybe Ambien, in regulating the stages of sleep. Are you aware of anything in that?
There are, the best evidence comes from other diseases like Alzheimer's and Parkinson's, where trials have gone on. Some light therapy, for example, is effective in Alzheimer's patients, as is some of the melatonin drugs. And my understanding is that the effects of light therapy are pretty similar to the effects of the drugs, but they're not big effects. Maybe, I mean, if you had to do it as a percentage, a 10 to 20% improvement in sleep. So they're not dramatic effects. And I've not tried melatonin-based drugs in the mice. They're quite difficult to use in mice, but I wouldn't expect dramatic effects from them. Unfortunately, I think the only alternative at the moment is to try different drugs and see which happens. I have heard of people who have opposite results from sleeping drugs, where a normal person might take a sleeping drug and it would put them straight to sleep, whereas I have had patients tell me that they've taken drugs that should have put their, or carers tell me that they have given drugs that should have made their patients sleep very well, and in fact have made them hyperactive. So I don't think we know enough about how drugs like this work on Huntington's disease patients. It's something that I think really deserves a good scrutiny.
Great. Yeah, I mean, I think that there's a number of different questions coming in, Jenny, about, do any of the sleep medications help HD patients? And I think what I'm hearing from you is, we just don't know exactly, and the studies yet have not been thoroughly done to answer that question, if there are medications out there that can be of assistance. I mean, with that...
Except, George, that there was a huge amount of research done on sleep in normal people, and not all of these drugs work the same in normal people. So at the moment, it is just trial and error, except there are a lot of drugs to try. So, and many of them are very safe. They've been tested very, very well. So if you are having sleep problems, you should see your doctor, and if they prescribe something that doesn't work very well, go back, because there are lots of other different sorts of medications, and some of these should help. So it's not hopeless, because the tests haven't been done. They've been studied very well in normal people, and there is a huge choice. So if one doesn't work, try another.
Great. Are you aware of any studies, sleep studies ongoing in humans that people can enroll in to be evaluated?
The only one I know of ongoing is in Cambridge that is ongoing, and so I think the enrollment is closed, but I don't know of any others. There have been proposals to have other sleep studies, but they're quite difficult to do, because to do polysomnography, you actually need people to come for 2 nights to stay in a sleep clinic, and you need to have electrodes. So if you have chorea, it's actually quite difficult to keep all of these electrodes on. So they're quite difficult studies to do in patients. So I don't know of any studies that are ongoing that people could enter, but I'm hoping that there will be some in the future. But they are quite difficult to do as well. So so they may take a wee while to get going.
Okay, and I have one more question. Looks like for you a scientific question, actually, Jenny. And the question is: would you hazard to guess as to whether the mutation is affecting retinal input into the SCN, the SCN itself, or secondary oscillators in the hypothalamus?
Okay, my guess would be that it's not retinal, that it's higher order differences. We have done studies where we've looked at some of the nuclei in the hypothalamus, and they actually appear to be pretty normal. So the inputs and their responses to input are pretty normal. So I think it's a circuitry disorganization that is causing these problems, both the sleep problems and the circadian problems. So I'd be very surprised if it was a retinal input, a direct retinal problem. I think it's a brain problem and probably a circuitry problem.
Great, thank you. And one last question, Jenny. Are there any specific type of doctors that you would suggest going to get help? Should patients and families just consult with their general practitioner or their neurologist, would you suggest?
With a neurologist, they may have a severe sleep problem. They may have a sleep problem that isn't related to Huntington's disease. So if you have a sleep problem, you should seek help for it, no matter whether you have Huntington's or not, because you may have a sleep disorder as well as Huntington's. And there are sleep specialists who understand the possibilities, the kind of medications that can be done. Not all neurologists will have a handle on the sort of details that are required, but many of them should be able to refer a person with a sleep deficit to a specialist, and that's what I would recommend. Talk to your doctor, find out how severe of a sleep problem you have, see if it's manageable with the drugs that anybody might take, and if it's not, see if you can see a sleep specialist who has a much greater understanding of the possibilities for treating a disorder that's as complicated as sleep disorders.
Okay. I have time, I think, for a couple more, if you don't mind, Jenny. In your human data you presented from the early on in your presentation, have you seen, or were you able to see if there's a connection to the number of CAG repeats with the dysfunction you're seeing in the circadian rhythm? Were you able to see a correlation there?
No. There's a correlation with severity, with time into illness, but we didn't look at the correlation between CAG repeats. We didn't have people who had very long repeats, so That's a really interesting question. It would be very interesting to know. I mean, I don't think juvenile cases have been studied in terms of sleep. No, so the answer is no. I don't know.
Okay, thank you, Jenny. I think there's maybe a few other questions, but hopefully we addressed most of them. If not, please email me. You can either reach me at gyorling@hdsa.org or just research updates at hdsa.org. And if Jenny or I'll email any questions to Jenny, or I will try to answer myself if we didn't adequately respond to your questions. And I'll just also throw out, I made a note to myself, Jenny made reference to the HD Buzz article that profiled good sleep hygiene. I think I actually have reviewed it. It's really a great article and a good resource. And what I will do on the HDSA website when we post Jenny's presentation next week, we'll also provide a link to that article so you can review that as well. So with that, I just want to thank Jenny again for a great presentation. Like I said, if you have any other questions, email us and we will get back to you as quickly as possible. Thank you, everyone. And thank you, Jenny.
Again, no, it's a pleasure. Bye-bye.
Bye-bye.